Hello? Are you muted? Yeah, I think you are. Can't hear you. Yeah, I can't hear. And it doesn't look like you're muted on the interface, so. No. Could be a problem with the sound card or the, when you logged in to the space. There we go. Jesse, how are you? Okay, Jesse's sound's working, so I don't know. I mean, you can type in the chat, too. Let's see, we've got some chat messages already. Okay. No mic today. Okay. Okay. I'm going to wait for a couple more minutes here for people to come in before I start. All right. So I hear someone breathing. Is that like... I think Jesse's muted, and I don't think Susan's mic is working, so I don't know where that's coming from, but... Oh. Yeah. Yeah, I still can't hear. Well, I think you can probably use the chat for just, I'll keep checking the chat, and we'll go from there. Oh, very low volume. Yeah, I don't know. Yeah, her sound card, I think, was having problems in previous meetings, so I don't know why it's not working. Okay. Welcome to the meeting. And we're going to... I changed the meeting venue because I'd already used this for another set of meetings. I like Jitsi. At some point, I'm going to try to upgrade it to something where I can control the interface better. Like Jitsi is an open source meeting space, and it works generally well. If you want to share your screen, there's if you want to do the chat, there's a button at the lower left of the screen. It's like a thought bubble. If you want to share your screen, there's a screen down at the lower left. And then there's a hand. You can raise or lower your hand. And that's all you really need to know for... Oh, Jesse, would you like to speak? I think it was just... hitting the hand. Yeah, it was okay. It was a hand raising. So that works. So you can see that in the corner there. And I guess if you set this up on a server, you can install custom elements. So that might come in the future. But for right now, we'll just use this setup. So a lot of things to get to today. We have some things on... I have some... interesting papers. I'd like to make some congratulations to Mayuk. I don't know. I was kind of hoping that Krishna would come to the meeting, and he was going to present, but he actually has some slides that he sent me on something he wants to submit to the Evolution Conference. We'll look at those, and then maybe we'll go over the task board. And so that's what I have for today. First of all, anyone want to bring up anything that they have from last meeting or from previous meetings that they wanted to revisit? Okay, they've been looking at the periodicity paper, a few edits, more to come later on. Yeah, the periodicity paper, actually, an update on that is we submitted it to Biosystems, and we already got a set of reviews back. So that was pretty quick. It's pretty positive. There are some small edits to be made as required by the reviewers. So we have some things like, well, there's probably some of the things that you hit on in the edits, but also some things that they wanted to change for, I assume, acceptance and publication. So that's good. We'll be revisiting those soon. I haven't gotten around to addressing them yet. Well, we might talk about them in the meeting if there's anything big, but I think it's probably all stuff we can just do on Slack or something like that. Oh, hi, Shruti.
Hello, hi, Brad. Hi, everyone.
Hello. And you have your camera on today.
Yeah, oh, yeah.
Well,
I read it.
Nice to see you.
Same.
Yeah. So we were just talking about the periodicity paper, so it's in pretty good shape. It's probably going to be accepted. We have to do some editing on it, and so we'll be talking about that in another meeting coming up. So, yeah. Is there anything you had, any announcements that you had from like past meetings or anything? Okay. So, so then let's move on. So we haven't done this year yet. I'll share my screen, and we'll do the major tasks for 2020, which is our task board. Can you see my screen?
Yeah.
Okay. So this has been a while since we've revisited this board. We have a bunch of things to do. We have get data for citizen spherical image data. I guess that's probably not a good way to phrase that, but this is something to do with the axolotl data. And I guess Susan said that there was more coming, and we have some that I have in my possession. So, is there an update on that, Susan? Oh, I have to check the chat. Okay, yeah, not yet. Well, that's fine. I don't remember when that issue was made. I think that was November 30. So I mean, when we actually did come up with a summer of code project for the axolotl data. So we might be able to get some people interested in that and move along on that. Though we already have data for it, to start off with. So it's not like we need to do it right away. It's not like we need to have it by then. Oh, there's Krishna. Hello, Krishna. You're muted, by the way. Okay. So, not yet. I'm working on the new microscope, but can get some older data too. That's good. Okay, so that's good. This build a collection of work for the Kindle book. So I think it was Krishna who asked if we could build a Kindle book of some content. I haven't really explored that yet. We can, if someone wants to think more about that, you're welcome to do so. but that might be a good way to sort of pull together a lot of the machine learning stuff that we've been doing and some of the other stuff. I don't know what goes into building a Kindle book. Maybe like just having like a electronic book that people can access, so they can access everything in one place. But that, I mean, I don't know how much work that'll take, but we'll probably get to that in the next couple weeks or so. As network science submission, this is something. There's a conference called CompleNet that is maybe a target conference. As I mentioned last week, we had I showed you the document of deadlines, which I think is in the Slack channel. So if you want to look that over, one of those submissions places is CompleNet, which is a complex networks conference, and we can submit something there. If we have ideas about networks in that area, we can submit there. And then this annotated bibliography. So we've been sort of talking about that over the past several months. This is still something up in the air. If people are interested in taking that on, let me know. We're thinking of doing this in my other group for a topic called developmental AI. So this might be, something where we'll have crossover with that. So in hold, we have updates on axolotl data and analysis. I think Susan kind of told us what that was. Recruit people as DevoWorm contributors. That's, again, that's something that we can do on an ongoing thing, but we don't have any events that will meet that goal. In the near future, probably something like this talk that I'm going to give at the OSF Unconference, and I'll tell you about that in a little bit. But other than that, this is just an ongoing thing. We also talked about tutorials for YouTube. So I think Ujwal actually made one last summer. And this goes along with the data science tutorials we have on the DevoWorm, in the DevoWorm organization. So this is something that we can do if people are interested in making a video tutorial. You can go ahead and inquire, maybe even make one, and then we can review it and put it up on the YouTube channel. So a lot of these things too, the embryo visualization, create embryo model in Blender, that's sort of a very long standing thing that we've been wanting to do, but to remind people, we wanted to build a virtual embryo in Blender, which is a 3D modeling software, and we'd eventually like to build something for the OpenWorm Docker, which is a software container that you can download and experience all of the OpenWorm simulations in one place. And we don't have a developmental version of that. So if we could have something that's developmental in scope, that would be great. So I think all these are just kind of, I don't have any updates on these. Update DevoWorm ML lectures. This is, of course, something that I was doing towards the end of last year. But we do have this DevoWorm ML course from 2019, and that content is in need of being updated. So if people have ideas about updating that, we can put that in this issue 73. This work on talk slides for OSF DevoLearn Flash Talk. That's something I'm going to get to in a minute. DevoWorm bibliography, that kind of goes with this annotated bibliography thing. That's something that we can update as we review papers and things like that. The bacillary psychophysics, that's something that we owe someone a paper on this in April. So we need to get back to this, but that's sort of on hold for now. But I mean, you can put it in hold, I guess, too. Let's see. Axolotl embryo animations and segmentation. That's something that, and it goes along with the axolotl data. So once we get that figured out, all of these issues can be addressed. The GSOC 2021 proposal, I think that's finished because we submitted those to Malin. And so now we wait and get people interested in making proposals for that. Recap presentation for 2020, that's finished. Great DevoWorm paper from Joss submission. That's it. Yeah, that would be an action item because it needs to be done, but it's kind of on the back burner still. I don't know, maybe hold for that. And then in progress, we have this DevoWorm bibliography, the Lagrangian embryo readings. I'm not sure. Oh, this is the thing that was a long time ago where we talked about a Lagrangian embryo. That's sort of on hold. Create a theory layer for DevoWorm. That's an on hold thing. Well, there's periodicity in the embryo paper. So that is actually, I would say that's an action item because the paper is pretty much, it's done and it's been pre-printed and we have this first set of reviews, but we just have to address the review. So maybe we can add a card here that says respond to peer reviews. And then that'll be in progress. And then this issue, when this is done, those two issues will be addressed. Then we have DevoWorm bibliography EndNote. I think that goes along with this other set of issues with the annotated bibliography and the bibliography. This is another issue. I think those are almost duplicates. Although this one, bibliography and EndNote, this is something that Dick and I were working on. And I think that part was done, but the other part about adding to it is not done. So, so we have complexity measures, which is an ongoing thing. That's where we, talk about different measures of complexity and development, and that's sort of an open-ended issue. And then this bacillary and non-normal cognition paper is the same thing pretty much as this one here. So maybe we should put follow-up on bacillary psychophysics is in progress. And that's basically addressing that paper. This issue is more about kind of following up on the measures. But I think we actually have that done, by the way, because it's something that was put into the talk that went to Neuromatch. So then we have these things that are off the radar. So these are things that we've had on the board that have been there for a long time, and I don't know where to put them. So these are things like there was a thread on computed tomography via cloud computing. That's in one probably about a year ago now that was brought up, and it never got followed up on. No one ever took us up on the offer to do anything. So that's off the radar, but if people want to address that issue, it's open. Krishna was going to review a paper. I can't remember what that was about, but that's something that Krishna, if you have want to follow up on that, you can put a note in this issue 44. This game theory for developmental processes. This is one of the open papers that we have in the open papers list. It's kind of been frozen in time for a while because it's kind of been doing a lot of other things. But if that's an interesting idea that, there's an outline that exists in that document right now, and it needs to be fleshed out more. And it might actually have some tie-ins with some of the things we're doing in my other group regarding developmental AI. So Jesse knows what I'm talking about on that. And, that's something that we can revisit maybe in the next couple weeks. There's actually a lecture in the DevoWorm ML materials called, it's part of the GANs lecture, or in that week, where we talk about game theory, because as it turns out, GANs are sort of inspired by game theory, and there's a interesting story there. But in any case, this might be an interesting thing to revisit. Follow up on neural organoids, number 30. I just talked to someone who I would talk to last year about a collaboration that involved building computational models of systems they call neural organoids. And neural organoids are these structures where you get a bunch of stem cells and you have them in like a 3D cell culture environment, and you grow them. You send you get them to differentiate into neurons, and then they grow these. They're sort of like embryoid bodies, so they have a certain shape. They're not really differentiated into a formal organism, but they're sort of differentiated tissues in a blob. And so they're using these as model systems for studying a number of things that neural systems do. And so I've been talking with a couple people about this over the last year and a half maybe. And it's been pretty low-key. I haven't really involved anyone else. But maybe the answer is to open this up. Maybe I'll talk about this a little bit more in future meetings. Basically, the idea is to sort of get our hands around a computational model of this, of neural organoids, and then maybe collaborate with some people who collect data in vivo systems where they're able to do experiments. So that's something that I think would be exciting if we could get it off the ground, but it's just been off the radar. This paper, this was something Jesse wanted to review at one time. That was something that was scale-free biology. That was a long time ago now, but if he's interested in that still, we can do that. There's also this compositionality paper, which is something that I don't think it's even in the open papers, but it's something that I was thinking about for developmental biology. It might be combined into this game theory paper. I don't really know. And then there's models for biological living networks. That kind of goes along with this network science submission. And so maybe I'll have more to say about that in coming meetings. So we've got... Okay. So now let's move on from that. I think if there are no... Well, okay, we have some things in the chat, first of all. So Jesse says, I'd be interested in the Kindle book. Okay, yeah, that's good. I'm going to try to get some... I'm going to try to maybe move on that a little bit. Or maybe put something in the Slack on this where we can organize our thoughts about how to put this together. I don't really have experience with Kindle books, so if someone knows how to do that, or if you could, we have to think of like a format to put things together, but we can do that. And then Susan's, now we have two S's. Yes, we have Shruti and Susan. So the Jitsi interface, when you have your camera off, has a little letter icon for your name. So Jesse says, RE annotated bibliography. I'll be interested in both parts of those, or in working on parts of it. Yeah, so like I said, there's a lot of overlap between this, like what we do in DevoWorm, and this initiative Developmental AI in my other group. And Krishna probably be interested in this too, where, we're talking about building a sort of an artificial intelligence system based on developmental principles. So there will be citations that'll bridge the two groups. So we'll see about that. I'm interested in measuring them with OCT. So interested in measuring what? I guess it was something organoids. Okay. The organoids thing. Yeah, it'd be interesting. There's some groups doing, like, you have to have an experimental setup, and I don't have access to that, or I don't know if Susan could do it because it's, it takes a pretty specialized setup. But, if we could find a collaborator, we might be able to do some things. And I know they do a lot of measurements with, some of the things that, like, voltage dyes and things like that, at least what I've seen in the literature. So there's a whole literature on measuring things in organoids, and there's room, of course, for improvement. And you can use a lot of microscopy techniques to do this stuff. So that's something we can talk about later. Jesse says, I forgot about the scale-free paper. I'd like to do it at some point, but yes, there are other things ahead of it for me. Okay. And then Susan says, I have a book I might want to put out as a Kindle book when you find out how to do it. Yeah, so we'll follow up on how to do that. I'm sure it's not difficult. I'm just making myself a note here. How to do and plan. So plan of attack, so we can figure out what'll be in it. And that'll go hand in hand maybe with the annotated bibliography or whatever. So we'll have this, at the end of the year maybe we'll have this nice collection of things that we can put out as a book, and people can use it as a reference. It's kind of like what happened with DevoWorm ML. That was something where kind of every week I did a lecture almost every week, and then I had other people contribute small lectures where they did like specialized topics where people brought papers to the meeting and we reviewed them. So if you go through the syllabus, you'll see that. I don't know, maybe that's a good way to do it. Have like a chapter where you delve into a topic a little bit, and I don't know, but definitely that's one model to do it. So again, the major tasks we review. If we want to add any issues, you can join the GitHub organization or join the GitHub repository, propose an issue or answer an issue there on GitHub. Okay, so the next thing I wanted to talk about. So Krishna, actually, I'll talk about your slides next. So you sent me a link to some slides on evolution, or the thing you wanted to present at the evolution conference. Okay. Yeah, there it is. Well, I'll bring up the slides and I'll go through them, and then you can tell me if what your comments are. So this is we're gonna do a talk here, or propose this talk. The winner takes all, or the winner gets killed. And so the first slide is, in an ecosystem does the winner. Oh, there is a problem with your mic. Yeah, it's a lot of background noise.
Hey, I am audible now?
Yeah.
Hello?
Okay. Yeah, it's a little bit better now. Okay.
Okay.
So yeah, this is the in an ecosystem, does the winner take it all, or does the winner get killed? So that's your question basically. So yeah, and then you have, I'm so confused. So what can you mention more about these three slides here?
Okay. So it's regarding the evolution conference that you were talking about in the previous meeting. So it discusses that. Can I share my screen?
Oh yeah, let me switch off here. I'll switch my screen off. All right, go ahead and share your screen now.
Okay. So here it is. Okay, so it's regarding the evolution conference. So in an ecosystem, does the winner takes it all, or the winner get killed? So here it is. Like for example, we are having two species that are competing with each other, and we say that due to some favorable circumstances, a species A gets it's get the edge over the species B. So for example, we have cats and dogs fighting with each other, and due to let it say that the temperature is favorable to the dogs or whatever be the reason, the dogs get an edge over the cat. So they started to their population starts to get increasing, and the cat population starts to decreasing. So here, if we put in terms of Darwin philosophy, survival is the form that will lead the most copies of itself in successive generation. So here the cats are are getting marginalized, and the dog population increases. So we can say that one day it will come that all the cats would perish, and we would have the dogs as the winner of this you can say fight. But the winner gets killed. So it was found in an eukaryotic. It was a study that happened with diatoms. So the winner get killed. So if you want to say that, okay, so if you say that there are two species A and B, and the A is, for example, the dogs are flourishing, so shall be the cats because they are preying on the dogs, and if their prey is flourishing, they should also flourish. So it's simple that if the both are trying to prey on each other, and it's simple that if one perishes, the other perish. If one flourish, the other flourish. So there is an equilibrium between the two of them. So it says that Volta-Volterra equation. It says that, for example, if if the population of mice increases, the population of cats also increases. Because cat preys on mice, but if the population of mice decreases due to scarcity of food, the cat population will also decrease. So it's in a relationship between the prey and the predator. So, but in practice, models show that the combination of competition and predation eventually made all the species go extinct. So, how it can be possible that this theory makes perfect sense and now suddenly with evidence we see that all the species that followed it are extinct. So, there is a new hypothesis by Nigel Goldfield and Cheese. It was so. What does it states that if we have two species that are competing with each other, and the one species tries to win, the population of the species increases. So does the population of the so does the population of the other species. It mutates in such a way that how can I put this into words, that it tries to invade its predator. So if the predators are getting bigger in population, the prey will also somehow evolve that it would be able to evade the predator, and so will be the predator evolving so that it can hunt the prey in a better manner. So this is a never ending cycle of improvement that helps every species to survive. So it's an initial draft of the presentation. So that's it. So the main thing is this, that co-evolving kill the winner theorem, and that normal kill the winner hypothesis.
Right. Yeah, that looks pretty good. Yeah, thanks for explaining. So yeah, I think that would be a good idea. And I don't know, if we want to, well, I mean, there are ways you can approach this. One thing is just to have sort of a theoretical approach. We might also consider using some sort of digital evolution approach, which of course we haven't talked about too much in the group, but involves where you simulate evolution in a program, and you can set up your problem so that you have your predator and your prey, and then you can simulate this. You could use something like Avita, which is a program that has specific parameters for mutation and reproduction, or you can use something like an agent-based model where I think you can specify the same parameters, but it's not as powerful. But in any case, I think we can address the core issue there, which is the hypothesis that you mentioned. We could do that, or we could approach it from a theoretical point of view. So just kind of like maybe updating the theory or thinking about how it applies to different scenarios or animal species. I mean, there are a lot of ways we can approach it. So why don't we? I'll think about it some, and you can think about it some. And so I mean, the submission for that would be, I think, either March or April 1st. I can't remember what it says on the.
So we have a lot of time to improvise it.
Yeah.
I think Dick Gordon would be very interested because the initial experiment was done on diatoms.
Okay.
And
yeah, that would be good. Yeah, there's the diatom theme again. So it's that would be great, and we could talk about that. Like the evolution conference is pretty broad, but there's certain sort of tracks it needs to fit into to get it to get it accepted. So we'll have to also think about it in terms of that. Like, what is the theme that it would fit into, or the track?
We can, change it into a theoretical portion if that's feasible, or we can, do some kind of experiment, as you are saying, that it would be something very interesting. And computationally modeling evolution is, I think, it would be a good task.
Yeah. Any thoughts about that or comments? I don't see anything in the chat, but if you have something, you can put it in the chat, and we can address it later. Well, thank you, Krishna, for that. I thought that was pretty good, so that's something that we'll be kind of making into a. We also have to make it into a abstract. But we should also. I like this idea of putting it in the slides first, and then, we can like kind of bounce back and forth between that and making the actual abstract, which actually helps you later because then you don't have to worry too much about making the slides. So, okay, so very good. The next thing I wanted to address here is major tasks. We've done that. I want to go to this figure for the boring billion, which is so this is something that I'm working on with Dick Gordon and George Mikolovsky. Actually, we have some comments. Okay, so this is the slides here for Krishna's presentation. Krishna and myself, his presentation, and then Jesse requested access. Okay, so let's see. So this is the. Just want to share my screen again. Okay, so this is the figure that I've been working on for this. So this is a paper that we're working on, and I was tasked with making a phylogeny of early life. And I did some research on this, and this isn't the flashiest figure in the world. I wanted to do a circular representation of a phylogeny, and of course a phylogeny is a tree of life where you start off with a single branch, and then life branches out into these different what they call taxa, or groups where that consist of either one or many organisms. And so that's the idea of this tree. And so as you can see, there are a bunch of things in this tree that so we start off here at 4 billion years ago. This is 4 10 to the 9th, that's billion. And so we go from that, and we look at the first, this is the first event where you get a division between bacteria and non-bacteria. And so this creates a common ancestor for bacteria, a common ancestor for non-bacteria. And then at about 2.85 million years ago, there was a split between archaebacteria and bacteria. And so archaebacteria consist of bacteria that are what they call extremophiles. So they live in ocean vents that are, hundreds and hundreds of degrees centigrade, and they can survive in extreme conditions. Whereas bacteria are the garden variety bacteria that exist on every surface at room temperature. And so there's a difference between those two groups, and they split here at this at 2.85 billion years ago. Then at 2.4 billion years ago, there was something called the Great Oxygenation Event, which is where you started to get oxygen in the atmosphere and in the rocks, in the oceans especially. And so you have this oxygenation event that occurs, and it kind of shifts. You know, the timing on these things are sort of estimated from a number of different sources. So we have estimates from molecular clock techniques, and we have estimates from fossil remains. And so the dates are not by any means like exact. There are ranges of dates, and I've kind of averaged out the different dates. So these dates might be off a little bit from, like, if you read something in one reference versus another, you know, the number might be a little bit different, but this is basically the gist of what's going on. So the boring billion is actually this time between like 2 billion and 1 billion years ago. And this is the sort of the basis of the paper here. And so what is happening in between those two times, and then what's happening before and what's happening after? So as you can see, what happens before the boring billion is not very much. What we have before 4 billion years ago is a lot of different, we have like, there are different hypotheses of what was going on at the time. There could have been things like RNA world, which is where you have a bunch of RNAs that are just freely living in liquids or on clay rocks. You have other things where there's no clear lineage, where there's a lot of recombination of things. So there are no lineages of different taxa, and we don't really know a lot about that period of time, but we know that life started somewhere before this point. And then at some point there was a split between bacteria and non-bacteria. Then you have this great oxygenation event, which is here, and then you get up to this Leka ancestor. So Leka, this is actually what they call Luca, down here at 4 billion years. And I didn't label it because I just forgot about the label, but that's Luca. And then this is Leka. So this is the one, this is the least, or the last eukaryotic common ancestor. Then Luca is the last universal common ancestor. So you see that there is a universal common ancestor between bacteria and non-bacteria, and then there's this last eukaryotic common ancestor between all these what they call eukaryotic organisms. And so all of these organisms have a number of different traits that define them. And so from 1.5 billion years ago, we have Leka. So we have this, now we have these eukaryotic cells. And so the difference between the eukaryotic common ancestor, well, we had the eukaryotic cells here, more or less. The difference between the bacterial cells and the eukaryotic cells, you can look into the literature on this, but the eukaryotic cells have a different they have different properties to them. But we don't really know much of what the common ancestor looked like, because as we can see here, at about a little bit after the time that we had this last eukaryotic common ancestor, we start to get things like a cell nucleus and photosynthesis. And so these are things that you'll see in plants and some photoplankton and things like that. So they're all, this is something that evolves relatively early, and doesn't exist, say, in animals, but these are things that evolve, and given the timing of these different events, it could very well be that this is around the same time as Leka. So the timing of this tree might be off a little bit in terms of what actually happened, but suffice it to say that this eukaryotic common ancestor, maybe had proto-versions of these traits, and then they are clearly recognizable in the fossil record around this time, or in the they can trace back the origin of this to this time. These are sort of like upper bounds to the existence of these things. So we know that, like, for example, we have evidence that we can date these things back to this time, but they may well have existed beforehand. We just don't know. And so from LICA, we have this split between protists, and then we have this other split, which leads to fungi, plants, and animals. So we still have this very general, we don't really have a lot of multicellularity yet down at LICA. We do get some multicellularity up further along here. I didn't put multicellularity on here, but what you do have, though, is you have this quick succession of events, and this is sort of at the upper bound of this boring billion. And what you have is you have this split between fungi and plants and animals at, I guess, 950 million years ago. And then you have this split between plants and animals at 850 million years ago. So those two things happen pretty quickly in succession. It's maybe not even clear in the literature, the order of these events, but they happened right around the same time. And so, but what's interesting is that you have this second oxygenation event, which the first oxygenation event, the great oxygenation event, is where you have this sort of the birth or this sort of first expression of oxygen in the atmosphere. The second oxygenation event is more extensive, and the level of atmospheric oxygen goes up quite a bit from its previous level. And so this enables a lot of innovation in these eukaryotic lineages and these different types of eukaryotic life. And so it's clear that from here, the data points to this sort of rapid diversification right around the second oxygenation event. And then I put the Doshanto embryo on here, and so I think last week we talked about this. This is the first evidence of an embryo in the fossil record. So plants have embryos. We haven't talked about that in the group, but the seeds of plants, they do have embryos that, it's a bunch of cells in an embryo. I had a professor once who called, a seed a baby in a box with its lunch. It's a little, group of cells that are sort of getting nourished by what's inside the seed. And the seed, because it gets dispersed, has a hard protective shell. It's kind of like an animal egg. And then it opens up and it allows the plant to colonize, new places or to plant itself in the earth and grow. And animal eggs kind of have the same function where they, you have, different animals that lay eggs. They can lay eggs and either, sit on them or leave them, and they hatch. And so they have this protective shell. And then eventually we get into placental mammals where they give live births. And so that's further up the tree here. But so this is the layout of the boring billion then is in between this sort of this area here and this area down here. So it's between the great oxygenation event and the second oxygenation event. And so what's basically going on there is not a lot on the bacterial side, although maybe there's a lot going on and we haven't figured it out yet. But I mean, what we're getting this sort of slow eukaryotic, maybe some innovations that are kind of coming together and maybe need to be enabled by the second oxygenation event, which then launches us into the modern version of life. And so that's something that I'm going to send to them pretty soon. And, I have some references here. So I didn't just pull those numbers out of a hat. I went through the literature on this, and it's not exhaustive, but I think it gives a broad outline of it. So if you're interested in this, I can send you a link to this document and share it with you. And I'm thinking of better ways to visualize this, but I don't know yet. Might be better to... you'd have to request access to it. I'm thinking of doing like a circular version of it, but I'm still trying to figure out how to space everything out. But I think this is basically the basic idea of what we want here. So, Okay, now I wanted to get to the, one more thing I wanted to show before I get into the other things here, the papers and that. So there's this simulation that I found online. This is really nice. And we've talked about like critical processes before. So this is it's like a long description of this. It has like relevance to viral pandemics, viral epidemics, and so this is it's something that demonstrates criticality on a network. And so this is a long sort of read, but it's a nice interactive application on the web. It's called Going Critical. Okay, yeah, thanks for the comments on the boring billion stuff. It's understandable the way it is. I'm wondering about that time scale myself. Yeah, we'll have to talk about that some more in future meetings. It's an interesting area. But anyways, I just posted a link on this Going Critical. And this is a nice interactive simulation / blog post by Kevin Simler. So they talk about networks, and they talk about diffusion on networks. And so this is an important topic for diseases, but also for memes, which are ideas. And you can study memes on social media, the spread of ideas, a wildfire breaking out across the landscape, neutrons cascading through a hunk of enriched uranium. So there are a lot of uses for a network diffusion model. And so he actually explores this in an interactive way. This is a simple model of a network. And in epidemiology, which he focuses on here, you have active nodes or infected nodes, and you have nodes that are uninfected or not active. And you have these edges whereby you can go from an infected node, and you can, and that infection can travel along the edge at a certain probability, so that all these four nodes that are immediately downstream of this node can be infected with a certain probability. And then if this one, say, is infected, then it can spread its infection to these four neighbors with a certain probability. And so then it can spread the infection through this network. And it depends on how connected each node is and the probability of infection, and that's how things spread. So this is how a disease spreads through a population. You have people, they interact, and they form these edges between themselves, and then they may infect their neighbor or they may not, or their people that they're interacting with, and then those people interact with a different set of people, they interact with those people and they spread, and so on and so forth. And so this network, it's not a linear model. It happens maybe in a linear way, but it has nonlinear effects. And there are things like cascades or, other types of phenomena that are, hard to predict using a linear model. You can also have critical phenomena. So if you have if this edge node, say, is infected, and then it spreads its infection to a node that is highly connected, such as this one, it can infect a lot of other nodes very quickly. Whereas this edge node, if it doesn't infect one of the more central nodes, then the infection dies out. And so this is kind of a way to think about network criticality using sort of an infectious disease model. And so this is where they put the network on a grid, and this is good for modeling purposes because you can figure out like the neighbors in the neighborhood. So we've talked about neighborhoods in the context of cellular automata, but here we have these neighborhoods of immediate neighbors and second order neighbors and so forth. And so it's a very good way to sort of visualize this process. And so something will propagate outwards from the center, and that should show the speed of propagation and, some of the critical phenomena. And so this is one of the simulations here. He's showing that an active node always transmits its infection to its uninfected neighbors in this way. So it's sort of a linear spread. And then, but this is dull, and then far more interesting things happen when transmission is probabilistic. So in this case, it's deterministic, and then in this case, it's going to be probabilistic here. So this is the transmission rate at 50%. So you can see there's this nonlinearity. If we go to like 23%, oh, okay, it didn't work. Yeah, okay, there we go. See, now it didn't really transmit much at all. Transmission rate of 88%. It transmits far more like the linear case. So now we find out that probabilities of infection of the neighbors actually creates this sort of asymmetric effect on this grid. And so you can play around with those parameters and then going critical. So now you have this, you think about the longevity of these things and transmission rates, and then this limitless diffusion has many names: going viral or going nuclear or going critical. So when something really breaks out and spreads all over the place, it's, we think of that as viral, but it's also it's a critical process, and that's more of the physics idea of it. You can have these things that are subcritical, which are these local expansions, but a critical expansion is where it just kind of breaks out and becomes a global phenomenon. So this tipping point is called the critical threshold, and it's a pretty general feature of diffusion processes on regular networks. So again, this is an example of something where there's some value, a critical value here. So if we reset it, we see that we start off with this little cluster in the middle, and it's kind of spreading out, and then at some point it just starts to spread everywhere. And it's maybe hard to see where that critical threshold is, but you can see that it changes its behavior at some point, and it becomes a global phenomenon. So it's kind of hard to interpret these things visually. Oh, this is the transmission rate on this. So this is 30% transmission rate. If we step it down to like 6%, okay, yeah, it kind of dies right out. If you go to we're at 16%, see, it dies out. If we go up to 43%, it's pretty much, it just goes global pretty quickly. 81%, yeah, it's just linear, just expands without any sort of anything to stop it. So this transmission rate then is going to determine how these things break out to being a critical phenomenon from a subcritical phenomenon. And so he discusses this a little bit more in terms of infection rates and epidemiological models and other things. And then he talks about spontaneous activation, which is sort of like, sort of a probabilistic thing, but this is actually interesting if you're interested in neurons, networks of neurons. And so the spontaneous activation rate, let's see. Okay, so it's not, yeah, it just kind of appears in different parts of in different parts of this grid. And it's, so you get like these infections in different parts of the grid, and then the transmission rate will determine whether these things take off. So you can see you had a couple spots where they start to pop up subcritically, and then eventually they take root, and then you get this expansion to a global state. Okay, so, and then this goes on for quite a while, and so I didn't want to spend too much more time on this, but I think it's a very good set of simulations for this idea. He talks about scientific networks and the diffusion of knowledge, which is, if you're interested in, like, even if you're interested in the philosophy of science, history of science, that might be good to check out. Also, if you're interested in epidemiology or even just networks, and like I said, if you're interested in networks of neurons, it's also very good to think about like how neurons propagate electrical signals across their network. Now, anyways, I think that's a good pedagogical tool. I would check it out. I put the link in the chat. And so the final thing I wanted to talk about today was this. I have a bunch of things in our papers queue. First of all, I'd like to congratulate Mayuk, who's not here today, but he just got accepted as an intern at Runway ML. And so this is a company, Runway ML. They're building the next generation of creative video tools with machine learning, and he'll be working on building the machine learning part behind Runway ML. and was thanking the people who were supporting him on this. So congratulations, Mayuk. I know you've been very good at... he was one of our summer code interns last year, and he did a lot of work with video in that project, and he's been working on a lot of GANs. So he's been doing some things, generative models of embryos and other types of patterns, pattern formation, and so this is very good. I'm glad to see that he's finding success, so congratulations on that. And then I wanted a couple other things I wanted to go through here. So a couple papers that we have, and again, if you have to leave at the top of the hour here, it's fine. Otherwise, we'll go on for about 15 more minutes on this. So one of the papers that I wanted to mention here, okay, we have a couple things in the chat before I get into this. Okay, so I have a couple. Okay, so for the periodicity paper, there are papers about the frequency of calcium concentration as an embryo develops. I was wondering if calcium periodicity could be studied alongside cell division data. Yeah, probably that could be a thing that we could look at or mention in the final version. I think I don't know how it would connect directly. I know there's some things about cell cycle and the timing of cell cycle in terms of genetic networks, genetic regulatory networks, but I'm sure that calcium periodicity probably also plays a role in that as well. And so I think we mentioned the physics of cells, so we might bring that up in the paper. And then Shruti asked, is the transmission rate the same as we call R0? Yes, that's very similar concept. The transmission rate is R0 on a net, but they're just showing this on a network or in a lattice of interacting people or interacting nodes, and they're just showing this rate of diffusion in the population. And I think R0 is something specific to epidemiology, where it refers to the infectability. So it's like a combination of the probability of being infected with your assuming a certain level of social interaction. So that is a very similar parameter. But the simulation I showed you largely just kind of makes us a little bit more abstract and shows you sort of the process behind these things, like how do things diffuse in a network. It could be disease, it could be information, it could be anything you can think of, electrical signals, whatever. The idea, it's basically a very similar process underlying all of that. So this is the first paper I have for today, what machine learning can do for developmental biology. And this is something that's very brand new in development. And this is a paper it's a review of basically for people, non-specialists in machine learning, people who don't attend this meeting. Developmental biologists maybe want to know a little bit more about what machine learning is all about, because you hear about it, and if you don't interact with the literature, then you don't know what it's about. So this is a nice review, I think. That developmental biology is grown into a data-intensive science with the development of high-throughput imaging and multi-omics approaches, which are like genomics, but they have phenomics, and they have other types of omics. They have all these next generation sequencing technologies, and so you can use all of those. Those are all part of developmental biology now. Machine learning is a versatile set of techniques that can help make sense of these large data sets with minimal human intervention. They're tasks such as image segmentation, super resolution microscopy, and cell clustering. In this spotlight, I introduce these key concepts, advantages, and limitations of machine learning, discuss how these methods are being applied to problems. I also focus on how machine learning is improving microscopy and single-cell omics techniques and data analysis. And then he provides an outlook for the future of these fields. So, yeah, he does a pretty good... This is Paul Villotry, who's a developmental biologist. I think he's interested in computation. He's a friend in France. I don't know what institution, but... he goes over a lot of like the recent developments in developmental biology, technical developments, focusing on the molecular scale and cellular scale. And then he kind of gets into the sort of the ways in which machine learning is applied. So he talks actually about some of the aspects of machine learning here. So you have, the things in developmental biology, and then you have these things in deep learning and in machine learning, like AlphaGo and other approaches to Go, which are games that machine learning has solved or has done very well on, I should say. And so then how do you integrate these? Well, one way is through microscopy. So you have microscopy techniques. You, of course, have image segmentation, which this group knows very well. Segmenting out cells, segmenting out membranes, segmenting out other things in microscopy images. This is something that's been done, for years using some computer vision techniques, various computer vision techniques. But now with machine learning, we can do this very quickly on a lot of images, and it makes it a lot more amenable to some of these large screens that they do in developmental biology, where they want to look for like different phenotypes for a bunch of mutants, and they'll take images of the maybe an embryo with different mutations, and they'll look for changes in the phenotype. And it's hard to do that without a method, systematic method for segmenting out features and comparing them statistically. And so this is one of the things that we can do with machine learning. He kind of gives us his own take on like the hierarchies of theory within the field of artificial intelligence. So it's like there's AI and then machine learning nested inside of that, and then deep learning nested inside of that. So, I mean, that's his approach. Maybe you don't agree, but and then so then the next thing is image resolution, which is where you can look at localizing individual molecules. So we haven't talked about that too much in this group, but you can use different molecular probes, fluorescent probes, to sort of visualize proteins and gene expression and other biochemical assay type things, and then identify these using machine learning, pick them out of a background. And this has been done traditionally either through like visual inspection or through some sort of thresholding. And so with machine learning, you can do this much more effectively. You can do label-free imaging, which is not labels in the machine learning sense, but labels in like amino, they call it amino-based staining, where you're staining for different things in the cell. And so there are other ways. That's another way another thing you can improve upon. And then data integration. So this is something we've been exploring in this group, where you can take different sources of data, like different types of things. Like, for example, we take like images of the cell that are sort of these bright field images, where you have just there are these gray images that we show with the cells, and then we have these fluorescent images where they have a black background, and the only thing you can see are these stained or these fluorescent parts of the image. And then you can take those two sets of images and merge them so that you have information. You can co-register essentially the cell, the geometry of the cell, with some marker that you're looking for, and put it into a spatial context. So this is something we've talked about in this group. And then he gets into the schematic of common tasks of machine learning. So there's classification, regression, ranking, clustering, and manifold learning. And all those things we can do with data from microscopy. We can use this approach, then we can use machine learning on single cell omics. So this is where we're looking at RNA sequencing in single cells, 3D conformation of DNA, of quantity of transcripts, which are like transcriptional output of different genes. You can use machine learning on these data, because these methods generate huge data sets, and so you need a way to like get through this data quickly, and machine learning lends itself to that. Now, we've talked about things like TSNE and UMAP, and he mentions them here under clustering. So these are methods that, and I don't know if some of you didn't see this talk. I gave it last December on TSNE and UMAP. So that's something that if you want to look at that presentation, those are different methods for sort of clustering data. A lot of times if they're building up a transcriptional profile of a single cell, for example, they'll cluster the transcripts in different ways and you can visualize them. And that machine learning helps on that process. Then he gets into deep learning and the inference of spatial and temporal relationships, which we talked about. And then he talks about growing an interdisciplinary community. So this is something we might revisit because they talk about how to foster this interaction between like computational methods, machine learning, and the developmental biology itself. And it's not easy because, our group is relatively unique in the world of this. It's a very small group of people who are sort of doing these things in parallel. And so, but we might try to think more in terms of community as well. And so, and then he talks about storing data, computational resources, all of that. And so this is a nice little perspective, and if you want to read it more about it, you can, I'll give you the link to the drive, put it in the chat. So ask for permissions for that if you need it. And so that's one paper. Another paper that I ran across this week was this, now we're shifting back to developmental biology and actually embryology. And this is a historical profile. This is Gavin Rylands de Beer, How Embryology Foreshadowed the Dilemmas of the Genome. So this is Gavin de Beer, who was an embryologist, and here's his picture. And he remembered at best as a shadowy figure among those who gradually built up our current view of evolution and the role of genetics. This view derives from the modern synthesis, the recognition that emerged in the 1930s, that genetics can adequately explain Darwinian evolution and speciation through natural selection. So before the 1930s, they didn't really have a good genetic basis for natural selection and speciation, but in the modern synthesis, which was kind of formed in the 30s, people sort of they were starting to build up a foundation, a genetic foundation for this. I argue that de Beer's theories of embryology had a crucial role in the modern synthesis, and that his work indirectly continues to influence how we think about the genome, evolution, and developmental biology. And so, so he was born in the UK, and he was educated in Paris. And let's see, his knowledge was encyclopedic. He was a gifted linguist. He was especially interested in Darwin. Let me get to the relevant stuff here. So these are the diagrams that Ernst Haeckel drew of embryos. So Ernst Haeckel was a 19th century embryologist, and he made this argument. He had this argument that basically said that that early embryos were similar. So if you look between lizards, tortoises, pigs, and humans, you see that there's a certain stage of embryogenesis where these all look very similar, right? Despite how different they are in their sort of final form or their adult form, they look very similar here. And then eventually they start to differentiate as embryogenesis proceeds. And so when you don't have a genetic basis for that, you think, well, there must be, they must all be basically from the same sort of origin, and then they have this developmental divergence here. But of course we know that their genomes are different. They're not that as different as you might think, but they're different enough that, that they're there like from the egg, and that they get expressed sequentially, and that even though they look the same here, they have different genomes and they're not necessarily from the same origin. So Haeckel sort of had this idea in the 19th century before we had genes, or this concept of genes and gene expression and all of this. And so this is the position that was widely held actually at this time. So the concept of recapitulation was something that they used to think. This was what this viewpoint of the embryo was, is that development recapitulates a lot of this sort of. So the recapitulation is like where they all basically have the same shape and then they diverge out. But of course, they didn't understand the mechanism underlying it, so they thought of it as sort of like a natural stage in every organism, and it wasn't. There was no underlying theory of what was going on. So they were misguided in that view. So basically, this person helped to correct this idea and ground it more in genetics. So now we understand why, you start off with an egg, you start off with these very similar forms, and you end up with widely divergent forms. And that it isn't just because they're all kind of from the same origin. We know that, there are a lot of things going on underneath the hood here. So the Haeckel embryogenesis had a solely phylogenetic meaning. This notion was captured in the catchphrase ontogeny recapitulates phylogeny. The scheme he proposed was Lamarckian. So Lamarck was someone who believed that behavior influenced evolution, and that was incorrect in terms of the Darwinian framework. And so in Lamarckian evolution, evolutionary novelties can only be acquired at the adult end of development, because earlier stages of embryogenesis were thought of to be the fixed adult forms of ancestral organisms, and so must be invariant. So this was something that was believed before Darwin in terms of how organisms change, and of course that was shown not to be the case. And so there was a lot of this idea that embryogenesis was preformed, even in the earliest stages. And so that was also proven to not be the case. So there is a lot of, this article really goes in, I think, into the historical aspects of how people used to think about evolution and development, and how that really changed over time, over about 200 years. And there were a lot of ideas in early embryogenesis that were, and this is true of any science, where you have early ideas that are incorrect, not because people were stupid then, but because they didn't have all the information that they would need to understand the system. So, if you don't know anything about genomics, if you don't know anything about some of the underlying biochemistry of organisms, then or even heredity, then if you don't have those theories and ideas in place, then it's really hard to make the correct interpretation. You make interpretations that seem correct. And this may hold true even for what we know today. We may not really have a full appreciation of what's going on in development, even given our knowledge of the genome. So these are some nice figures. If you want to look this paper over, there are a lot of nice figures that show these sort of processes. So there's the preformation hypothesis, there's the one-step transformation hypothesis, there's genetic preformation, and then there's epigenesis, which is currently the orthodoxy and makes sense given what we know today. So epigenesis is where you start off with this bag of genes and a single cell, and then you get differentiation of genes or of cells, and you get changes in shape, and then you end up with this frog. Before that, of course, we thought maybe that there was some sort of preformation that went on, or that it was instant transformation from a common form. And so those all were proven to not be the case. And so again, here's this is sorted by thinker. So now he gives an example of how different thinkers sort of thought about development. So von Baer, who preceded Haeckel, thought of development in a certain way, and he had certain assumptions. And then Haeckel came along and changed that a bit, but it was still incorrect, at least the way we understand it today. And then now we have this revised version, which is where we're thinking about this explicitly in a phylogenetic context. Although Haeckel had a phylogenetic approach as well, it was based on these changes happening as you go along the span of development, so that every embryo was basically from the same sort of source material and then got shaped in development. Whereas nowadays we know that there's this evolution of development where evolution constrains development, and then there are changes in development, but they're not due to like this sort of preformed view of the world or preformed view of organisms. So it's there's some of these differences are very subtle, but they're important to recognize because when you put together a theoretical argument, sometimes it's hard to make these distinctions and understand the history of all this. So I would definitely take a look at this paper if you're interested in the sort of the history of biology, but also the history of how theories are formed and other things. And so this also brings us to this era of genomics, in that in our current era, we have a lot of data about genomics, but we don't really understand the sort of what's going on underlying the data. And so in one of his last publications, Homology and Unsolved Problems, De Beers summed up these issues in a masterly fashion. He argued that genes are no more reliable than adults or embryological characters as measures of homology, which is like the similarity between different species. What applies to embryos in adults must apply to the genes that determine them. With the advent of molecular genetics, we now know how right he was, and that different DNA sequences change at vastly different rates. So which genes should one choose as a measure of evolutionary change? And how should data from the collectivity of genes in a species be put together in such a measure? And so we know a lot about gene sequences and their expression levels, but we don't really know how they sort of contribute to this form. And so that's really one of the lessons of this paper, is that they talk, they kind of put that framework in place for you. So I think that's good. Let me look in the chat again. So Susan said mechanically similar processes, vertebrates. Okay. So, that's, yeah. So that's, yeah. So, there, if you want to follow up on some of these papers that I've shown, it's great. Or some of the issues that we talked about today, that's great. So, yeah, I think that's all I had for today. Thank you for meeting. Next week, maybe we'll talk more about some of these other submissions. I didn't even talk about the flash talk for this OSF conference. I didn't get into those slides. I don't really have them organized yet. But what we need to do for this is, I think, create a 5 to 10 minute talk on DevoWorm. And DevoWorm, the platform and how it might be used in education and getting our ideas across in the group. So I think that's something that we'll do next week. I'm going to work on it some. The presentation for that is on February 7th, and I'll send some more information about that event closer to the date. They're going to have a lot of stuff on online education, so if you're interested in that, you might want to attend the unconference. It's on, I think it's on like Zoom or something like that, and I'll get the slides in better shape for next week, and we'll go through them. It's basically, though, just going to walk through DevoWorm and how it's being used, how it was made, and then our kind of a vision for it. And then maybe we can go through and think more about like how, this might be used in our group and to recruit people and educate people on these issues. So, anything else before we go?
Actually, I wanted to show the general biological code, but I think that we are, running out of time. So maybe the next time.
Okay, so this is like a...
Okay,
is this code or is this like a...
It's the code. I was talking about the general biological models.
Okay.
So it's the code regarding that. I'll send you the Google Colab link. So you can see it. In the next time I'll explain that how the things are going.
Okay, that sounds good. Yeah, and we can walk through it. Yeah.
Yeah.
Well, I'm glad to see you're making progress on that. That's very good.
Yeah.
Okay. Anyone else have anything before we go, or? Got another comment in the chat. Okay. Oh, yeah. So Susan followed up on the vertebrates develop around a beating heart. So this is, this had to do with the paper that we talked about. Yeah. And so, Susan, if you have any papers that you know of that might be relevant to following up on this paper or some of the other stuff, why don't you send them along in an email, and we'll do them next week. That'd be good. Jesse says thanks. So thanks for attending. Have a good week. Talk to you later.
Yeah. Bye.
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 Bye.